Hello class, this is where we left off of last time. We actually were just on the slide before this, which was our start of our Krebs cycle, where we were just getting our basics down. I was going to have you guys watch this video just to remind us about the whole presentation, the whole cycle, I guess, before continuing. So this is still under the Krebs cycle. There's our first step, which is we also need to get our acetyl-CoA formation. So the Krebs cycle has a few steps within it, but we also need our acetyl-CoAs. So our input is an acetyl-CoA. Sorry, let me start over. Our location is still the matrix of the mitochondria. It's still in this part of our citric acid cycle, our Krebs cycle, but what we need to do is take our two acetyl-CoAs, which were created in our first few steps. We'll talk about the details of that. And then a lot of 6 NADHs, 2 FADH plus, and 2 ADPs. And then we'll create some more carbon dioxide, 6 NADH, 2 FADH, and 2 ADP. So the way this works is we convert two pyruvates into acetyl-CoAs by having NADH plus remove an H plus, becoming NADH. So we are creating our acetyl-CoA, which is shown right here, by removing an H, a positive proton, so that NADH can now become NADH, or sorry, NAD plus can become NADH. This creates two of our carbon dioxide molecules, and now we have a 4-carbon molecule from earlier bonds in our citric acid cycle, from an earlier cycle. So since this is a cycle from our beginning of the last cycle, we have a 4-carbon molecule which will bind to our acetyl-CoA, making our citric acid, which has 6. Once we take our 4-carbon molecule from an earlier cycle and combine it with our acetyl-CoA, we'll have a 6-carbon molecule. Then an NADH plus removes an H plus from our acetyl-CoA, making NADH, CO2, and a 5-carbon molecule. So we're just going around our chain. We were just here. Now in this step, So we are at this point where we have a new 5-carbon molecule known as this alpha cataglycerate. So our next step would be to create some more NADH, and this happens when NAD and ADP remove an H plus and a phosphate group from this 5-carbon molecule to make NADH, ATP, and some more CO2. So we just keep going around this circle. This 4-carbon molecule now is rearranged by the enzymes and produces 2 NADH and 2 FADH2. And then we have a 4-carbon molecule which is used in our next cycle up at the top. Alright, so the third step in our the part of aerobic respiration is the electron transport chain. This is very similar to the end of photosynthesis. This takes place in our inner mitochondrial membrane. So if we look at where our mitochondria is, the inside is our matrix, then we have our inner membrane right here, inner mitochondrial membrane. We're going to put in our 10-ADH that we just made, our 2 FADH2, and this is where we will use our 6 oxygen. We'll also need 34 ADP so we can get 34 ATP out. We'll use our other batteries and produce some water. So if we look at that in detail, we have our NADH and our FADH2, which bring our hydrogen protons and electrons to the hydrogen pumps. So the way this works is our energy molecules bring these H pluses, or bring the energy right here, which allows the H pluses on this side of the membrane to be moved across these protons into a large gradient. Now, naturally, once they're all here, since there are more on this side of the membrane than on this side, they will want to find what's called equilibrium. They will want to be equal, where we would have maybe 5 and 5. So since it naturally wants to do that, they will go through this this enzyme called ATP synthase. But our electrons are used to help move this H plus in, then they flow back through ATP synthase, and ATP synthase will take NADP and create NAD, or I'm so sorry. It will take ADP and create ATP. So then we will have, what happens here is we have a lot of extra H pluses floating around and extra electrons that have lost their energy. So we take our oxygen and make water. So this is where our oxygen gets put in. It's to absorb some of these H pluses that are coming through and these electrons, and also water. So a summary of this, I know there's a lot of maybe confusing parts, is that glucose is broken down into pyruvate. Pyruvate is changed into acetyl-CoA, creating a lot of NADH and FADH2. The electron transport chain takes NADH and FADH2 to create a hydrogen gradient, and the hydrogen gradient is used to create about 34 ATP. So our final input and output that I would like you guys to have in your notes is that our glycolysis step, we take glucose, 2 NAD+, 2 ADP, and 2 ATP, create 4 ATP, 2 NADH, and 2 pyruvate. Then that pyruvate is used in our Krebs cycle step, along with 8 NAD+, 2 FAD+, 2 NADP, and we will produce 6 CO2, 8 NADH, 2 FADH, and 2 ATP. In our final step, we'll take all of our NADH and FADH2 that we made, use 6 O2 and 34 ATP to create 34 ATP, 10 NAD+, 2 FADH+, and 6 H2O. So these ones that I have bolded out are actually the molecules in our overall reaction, which we had at the very beginning outlined. So the rest of these will actually cancel with each other. So we have 2 pyruvate and 2 pyruvate. So we will subtract 2 pyruvate from 2 pyruvate. So they will cancel out, and we'll end with our overall reaction that we wrote at the beginning of our lecture. So I hope especially this part, these last 2 slides, are the very, very important things to take away. And I hope the extra videos have been helpful as well to help explain this somewhat difficult process. So if you have any questions, please let me know.